Welcome back to our channel. Today we will dive deep into an essential topic in microbiology, the media types and nutritional requirements of microorganisms. This video will be particularly helpful for students preparing for exams. By the end of this session, you will have a strong understanding of the different type of culture media and the specific nutrients needed for microbial growth. Let's get started. Microorganisms require a variety of nutrients for growth and metabolism, broadly classified into macronutrients, micronutrients, and growth factors. The macronutrients, including carbon, nitrogen, and phosphorus, are needed in large amounts. Micronutrients like iron and magnesium are required in smaller quantities but are essential for enzyme functions. Growth factors such as vitamins and amino acids are required by some microorganisms that cannot synthesize them on their own. Additionally, based on their carbon and energy sources, we classify microorganisms into autotrophs, heterotrophs, phototrophs, and chemotrophs. Now let's explore the different types of culture media used in microbiology. Media are classified based on their consistency, composition, and function. Liquid media, like nutrient broth, are used for growing bacteria in bulk, while solid media, such as nutrient agar, help in isolating pure colonies. Defined media have a precise chemical composition useful for studying nutritional requirements. Complex media, which contains ingredients like peptones and yeast extract, are ideal for growing a wide range of microorganisms. Selective media, like Maconke agar, are designed to suppress the growth of certain bacteria while allowing others to thrive. Differential media help in distinguishing bacterial species based on visible differences, such as color changes. Blood agar is a great example, helping detect hemolytic bacteria based on their ability to break down red blood cells. Let's explore some special types of media designed for unique purposes. Transport media, such as stewards and amines, are used to preserve samples during transport from the clinic to the lab without allowing microbial growth. This is crucial for accurate diagnosis. Anaerobic media like thioglycolate broth are designed to grow anaerobic bacteria by reducing oxygen content. Chromogenic media are advanced tools that contain substrates changing color upon reaction with specific enzymes. For instance, chrome agar is commonly used in clinical labs to quickly identify Candida species based on distinct colony colors. To ensure successful microbial growth, the preparation of culture media is critical. First, all the ingredients are accurately weighed and dissolved in distilled water. The pH is adjusted to an optimal range, usually between 7 to 7.4, which is ideal for most bacteria. The medium is then sterilized in an autoclave at 121 degrees Celsius for about 15 minutes to eliminate any contaminants. After cooling, the media can be poured into petri dishes for solid media or stored in flasks for liquid media. The growth of microorganisms in culture media is influenced by various factors such as temperature, pH, oxygen levels, and osmotic pressures. Different microorganisms have specific requirements. For instance, psychrophiles thrive in cold temperature, while thermophiles prefer high temperatures. Similarly, some bacteria, like aerobics, need oxygen to grow, while anaerobics cannot survive in its presence. Culture media play a vital role in various practical applications. In clinical microbiology, they help isolate and identify pathogens. They are also used in antibiotic sensitivity testing to determine the effectiveness of antibiotics. In research, defined media are utilized for studying metabolic pathways. Additionally, in industrial microbiology, media are used in fermentation process to produce products like antibiotics, enzymes, and vitamins. As you prepare for exams, focus on understanding key definitions and differentiations. Questions often revolve around examples of selective and differential media, their applications, and factors affecting microbial growth. For instance, you might be asked to explain why McConkey agar media is both selective and differential. Use practical examples to illustrate your points. Reviewing this concept and practicing questions like this, like those listed here, will help you gain confidence and improve your understanding. In conclusion, understanding the various types of media and their role in microbiology is key for both practical lab works and exams. Focus on differentiating between media types and their specific uses in clinical and research settings. Don't forget to revisit this topic during your exam preparation and make sure you understand the key differences and applications. If you enjoyed this lesson or the video, give it a thumbs up, subscribe for more, and check out our channel for additional resources. Happy studying and best of luck on your exams.